Good afternoon, everybody. Welcome to Lockdown Bioinformatics, number 44. Yes, welcome back. And we're going to pick up where we left off from last time, as we always do. Last time, which was 2 days ago now, because we, if you remember, our new regime, we're doing this 3 times a week, not 5 times a week. So last time around we were talking about our new bit of work that we're going to do, and that new bit of work essentially is to look at the alignment of short reads to a reference genome. And what we did on Monday in lesson 43 was that we downloaded some data that we were going to use for especially for that purpose. So we downloaded some data, some short read data that we were going to align, and we downloaded a reference genome to which we were going to align it. We started doing that. We did that at the command line, and we set up a new Conda environment for us to do that from, and got ourselves to a point where we had all of the data that we're going to need. And it's today that we're going to look at how to actually do that alignment. And then what we spent the rest of number 43 talking about was this data transformation concept called the Burrows-Wheeler transform, which is used by a lot of these alignment protocols to transform the reference data so that it enables quite a few things. It enables it to be stored in a compact manner, and also renders it more searchable. And that was the key thing about this transformation, is that it makes searching for matching sequences in your genome based on your reads, it's a much easier process than it would be if you didn't transform that data in that way. So I spent some time going through the process of transforming string data in this way. So we looked at how the data gets rotated. So you get this set of rotations of your initial input string, which are then sorted alphabetically. So that's sorted by this first column here lexicographically, and then it's this final column is what we store, and is the Burrows-Wheeler transformation of our original string. And then I did go on to show how having the data stored in this representation, along with the information that's in this first column, about how many occurrences of each type of character there are, having that information stored in this index renders this much more searchable representation of the string than you would have had otherwise. So searching for instances in a string this simple is relatively straightforward. So we looked for instances of this string ANA. Obviously, if I was just doing that by eye in our original search string, that's a relatively straightforward thing to do with such a simple thing to search. You know, that our genome here is very straightforward. It's only got 6 characters in it. But in a human-sized genome, so a 3 billion base pair genome, searching for short strings in such a large database is a tricky thing to do, and the Burrows-Wheeler transformation makes that much more straightforward, makes it easier to do that. So that was kind of the subject of lesson number 43, and what we're now going to do in this one, in number 44, is to apply some of these techniques. And luckily we don't have to do this from scratch. We have software available to us that apply these transformations for us and then use that transfer for searching for occurrences of our short reads in our transformed genome sequence. So we're going to use one of those pieces of software, and the software that we're going to use is called Bowtie2, which many of you may already have heard of. And Bowtie2 uses the Burrows-Wheeler transformation and applies it to the reference genome of interest. So transforms that reference genome according to this this regime that I illustrated last time out, which makes it nice and searchable. And then you search that your transformed reference genome, but you search it not with the whole read. So you don't take the whole read and search for an exact match for the whole read, because that doesn't allow you any flexibility. So the way that you search the Burrows-Wheeler transformed representation of your genome doesn't allow for things like gaps and mismatches and stuff like that. So, which are all desirable when you're doing a DNA sequence alignment. So you do what's called a seed match. So you take a small section of your read, and you use that small section of the read to search the Burrows-Wheeler transformed genome to find candidate matches. So to find candidate regions where you may have a match to your read, and then all of those candidates are then expanded using a slightly more refined alignment algorithm. So Bowtie2 in particular uses the Smith-Waterman algorithm, which is a pairwise sequence alignment algorithm, dynamic programming algorithm, which goes back to the 80s. It's been around a long time, but that's then used to enable you to find the best alignment, the best overall alignment for your read sequence to your reference genome. So that's the process that we're going to follow with short read alignment. So the first thing that we have to do to enable our short read alignment is to use this Burrows-Wheeler transform to make a genome index. So given a genome sequence as input, we want to create this Burrows-Wheeler transformed index of that genome. So we're going to use Bowtie2 to do that. In order to do that, we need, first of all, to install Bowtie2. So let's begin there. So let's go to our virtual machine, because these are all command line tools. So we're working back in command line Linux like we were in the early lessons. So let me get rid of that. There we go. And here's my virtual machine. So my virtual machine is still running from the last time out. We downloaded the data last time around, so we used prefetch from SRA tools to download the appropriate SRA file. So this, on the screen here, you can see the accession is here. So SRR 118666736 is our accession. So we used prefetch to download the SRA file, and then we used fastqdump, and you can still see the command here on the screen. So fastqdump with a few options to convert that SRA file into FastQ files for our kind of follow-on purposes for the things that we're going to do next. So after doing those two things, we've got three files. So we've got a directory which is named after our accession, and inside that directory we have the SRA file, which is the result of the prefetch command. Have we ever looked at an SRA file? I don't think we have we? Okay, let's have a little bit of a diversion look at an SRA. So I want to talk a little bit about data transformations and data integrity as well. So I'm not sure, when we downloaded our RNA-Seq data, I don't think we ever kind of detoured and had a look at the contents of an SRA file. I know that we've seen what a FastQ file looks like, but I don't think we've ever actually looked at the contents of an SRA file. So let's have a look, because it's like our FastQ files. In the end, an SRA file is text, so we can have a look inside it using our standard text tools. So we're going to use less, which is if we're going to really refresh our command line lessons, then let's remind ourselves of what less is. It's a program for viewing text files a page at a time. It's called a pager application. So if we use less to look at our SRA file, it looks like the SRA is binary, which is interesting. Didn't expect that. Okay, so it's binary, so it's going to be garbage. I wonder how that might be compressed. So let's try and figure this out. It just says data. Z-less? No, that doesn't work either. Oh, that's a surprise. I expected the SRA file to be text. And I'm not sure why it isn't. Okay, I might have to row back on my suggestion that we were going to look at the data inside the SRA file. Using SRA, I'm sure that we can get the SRA format out of it if we want to. I would imagine that there is an SRA tool for doing it as well. SRA tools. Is there a view? I don't know. I'm going to have to row back on this. Maybe I'll do this another time. It was a nice idea, but let's do this another time. Okay. So an SRA file. My experience of SRA files is that they are they have text in them, and that text is in a very different format to the FastQ, which is going to be the point that I was going to try and illustrate by looking at the SRA file. Was that the contents of that SRA file is very different to the contents of the FastQ file, and that in order to convert it to FastQ, quite a lot of work has to be done, and that actually you're relying on the annotation in the SRA file to reliably convert that data into FastQ. So one of the things that I've experienced in the past is that in the SRA file, the data is recorded as spots. So one spot, which is one cluster on the flow cell, is recorded as a single kind of data object. And that spot, in the case of a paired end experiment, which we have here, actually contains a pair of reads. But in the SRA file, they're concatenated into a single piece of data. And then there's information, there's metadata in the SRA file, which tells you how to split that read into two. So usually you split them evenly. So you might have 100 bases in the SRA file, which is supposed to be split into two 50 base pair reads. And that split is encoded in the metadata that is within the SRA file. If that metadata is wrong for whatever reason, then you can end up with unexpected results. So I have seen an example of exactly that, a 50 base pair paired end experiment stored on the SRA, where the metadata in the SRA file was wrong, and when you used FastQ dump to split it out into its paired end reads, you ended up with one 70 base pair read and one 30 base pair read, because the metadata was inaccurate and so that read wasn't getting split correctly. Now what I'm going to assume for this experiment, and I've not had a chance to check, is that that hasn't happened. We've got decent data in our FastQ files. That's a little that failed completely, so sorry about that. But we do have FastQ files, and we know that they're gzip compressed text. Because it's paired end data, and we told FastQ dump to split out the pairs, so we end up with two files. In each of those files, we have one of each of the pairs, and it looks like. So we can see a 71, but then a 250 and then some 251 base pair reads. So it looks like these might be 251 base cycle reads I know from the metadata on the SRA that it's a MySeq experiment, so that's capable of producing these 250 base pair reads. It's actually capable of doing 300 base pair reads. So we've got some nice long reads. That's our first in pair reads, and in the _2 file we have the pairs of those reads. So this is the second in pair reads of those same spots. So that's our reads encoded in FastQ, which is the way that we want them for using Bowtie2, which is what we're going to use for our alignment. We've also, the other thing that we downloaded in the last session was our reference genome. So we downloaded the E. coli K12 reference genome from the NCBI. That is what's in the other file that I've got here in my week 10 directory. So this file called GCF_00058 blah 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 blah. So this file, which is also compressed using gzip, so I'm going to use zless again to look at the contents of this. This is the E. coli K12 reference genome, so about 4.5 megabases, I think, in the chromosome of E. coli. So we've got our chromosomal DNA, and then it may well be the case that there's a plasmid or two in here as well. So we can use grep to find the header lines in our FastQ. So lines which begin with greater than. So let's use grep to pull those lines out from here, just to see how many sequences there are. So it is just the main circular chromosome. That's all that's in that reference genome. So just a single sequence. Okay, so that's our reference genome, and we've got our reads. So what we're going to do first of all is we're going to transform that reference genome using the Burrows-Wheeler transformation. And the way that we're going to do that is by doing a process called a Bowtie process called build. So we're going to use Bowtie2 to build an index for our reference genome. Before we can do that, we need to install Bowtie2. And so we're going to install it in the same way as we've been installing all of our bioinformatics software so far. So we're going to use Conda to do that installation. So Conda, I'm in my alignment Conda environment, so you can see my Conda environment over here on the left hand side. If I wasn't in the correct environment, so in my other terminal here, you can see I'm in the base environment here. So if I'm in the base environment, then I need to Conda activate my alignment environment to get into the right environment, and then Conda install Bowtie2. Okay, so Bowtie2 is the software that we're going to use for our high throughput sequencing alignments. So we're going to ask Conda to install Bowtie2 for us, and it's going to do what it always does. It's going to try and set up and resolve its environment, find the appropriate packages and dependencies that it needs to install for us to have a functional installation of Bowtie2, which is that list of things there. And then it's going to install those pieces of software to give us access to Bowtie2 as a piece of software. So we're looking at installing here Bowtie2 version 2.4. So this should take a couple of minutes. The other thing that I want to do actually while we're at this, while we're doing software installation, is in this environment I want to install FastQC and MultiQC, because we should never embark on any bioinformatics process with sequencing data without first doing some QC. So we need to continue to kind of reinforce the lessons that we've learned before, and we should be installing and running quality control at the same time as doing any of these bioinformatics processes. So we shouldn't skip that just because we're trying to do something new today. So once the Bowtie2 is installed, I'm also going to install those QC packages as well, so that we can do we can have a look at the QC of this data as well. So Conda's just going to take a couple of minutes to work all of this stuff out of the system. While Conda is running, I will just quickly summarize what we're going to do today. So back to my whiteboard. So install aligning reads against the reference genome is essentially a two-step process. So we need to build a genome index. I can't spell. Build a genome index. So to do this, we use this program which is installed with Bowtie2 called Bowtie2 build. That builds the genome index, and then we align. My writing has gone to pot. Sorry. Align reads to index, and this uses the binary which is called Bowtie2. Okay, so that is our kind of two-step process. The thing about Bowtie2 and actually pretty much any high throughput sequence aligner, so this is true for BWA, it's true for RNA-Seq aligners like STAR as well, is that the process of building the genome index, that's a relatively kind of taxing process. It's a relatively intensive process to undertake, and what you don't want to be doing is sticking this onto the front of every alignment that you ever do. So you don't want to have to build an index to align against every time you want to align some reads to a reference genome. And that's why building the genome index is a standalone process, because it's this intensive process that can take some time. And actually with a human-sized genome, just building the index takes quite a lot of computation. You want to do it once, and you want to store that index. So you're going to store this index on your disk, rather than so you don't want to have to compute that on the fly every time you align reads to the index. So then Bowtie2 takes that index as input. So that's part of the input for Bowtie2 is the index that you build in the first process. Okay, so that's what we're going to do over the next half an hour or so, is this two-step process. So let's have a look at how Conda's getting on. Okay, so Conda's done for Bowtie2. So the other thing that we wanted to do was to install our QC tools in this environment as well. So Conda's installed these tools before, so it should be a case of just linking them to this environment. We're going to install FastQC, and we're going to install MultiQC as well. So there's FastQC. I suspect that the reason that Conda wants to install all of these things is that there's updates to all of these dependencies. So it's reinstalling a load of things here because it's getting all of the updates to these dependencies. So things get updated over time. So you see, despite that long list of dependencies here, it's actually only downloaded and installed a few things. They will have been the dependencies that have been updated since the last time I installed FastQC. So the other thing that we want to install is MultiQC. So let's do that as well. And then we've got all of the tools linked to this Conda environment that we need for our subsequent processes. So this is the last little installation. So again, a long list of dependencies and things to install, but only a few of them are actually being installed. Interestingly, MultiQC is now at version 1.9. I would have expected that to be... Oh, I feel like it should be downgraded. Okay. So there's a new version of MultiQC since we last ran it. I know that. There was a new release of MultiQC, I think just earlier this week actually, version 1.9. So consequently, Conda here has quite a lot of work to do because it's got to make sure that all of those dependencies are up to date for that new version of MultiQC. So it's just dealing with all of that. If you ever want to update a piece of software which you've installed with Conda, then Conda comes with one of the commands for Conda is update. So you can do Conda update, and I think if you just run Conda update with no arguments, it will update all of the currently installed packages, or try to update all of the currently installed packages to the most recent version. Or you can name a piece of software that you've got installed. So Conda update MultiQC will just try to update MultiQC. If you're running the current version, it should say that you're all up to date and there's nothing to do. I hope that's what's going to happen now. So you can use Conda for updating software that you've got installed. Okay, Conda's being a little bit of a pain today. I don't know why. I never entirely understand the inner workings of Conda, if I'm brutally honest. Okay, that's going to do some things, and that's fine. But that it can be nice to make sure that we're running the latest version of things quite often. And obviously, as I've mentioned before, you can have different versions of software installed in different Conda environments as well. So there you go, that's done. Finished doing whatever it was trying to do. Right, okay. So that's got us all of the software that we need. We've got all of the data that we need. And so now it's a process of running the appropriate software over the appropriate inputs. So the first thing we're going to do is run FastQC. Okay, so FastQC, if you remember, and you may or may not remember, so we can look at the help file if you don't, but FastQC, the simplest way of running it is to give it a list of FastQ files as input. So if we go up to the synopsis at the top of the help file, then the simplest way to run FastQC is with a list of inputs. So a list of sequencing files. So that's FastQC, and then any FastQ files that we might have in our current working directory, we can run with just star.fastq.gz. We'll list those files for us. So this will run FastQC over those two FastQ files. And again, this will take just a minute or two. Hopefully it won't take an overly huge amount of time, because these aren't, although this is a proper full-on experiment, these aren't massive sequencing files because it's bacterial sequencing. So it takes a big experiment for these sequencing files to get big with bacterial sequencing. I think there's about 1.3 million reads or so in each of these files, so it will take a couple of minutes, but it won't go on forever. So this is running FastQC, and FastQC produces, if you remember back to the appropriate session, which I think was about number 13 or so, maybe a little bit earlier. In fact, maybe a little bit later, actually. We introduced FastQC and we looked at the reports. There you go, we look at the results of FastQC. That was number 18, in fact, was when we looked at the results of FastQC. And the one before that, session number 17, we looked at running FastQC and running MultiQC. Admittedly quite a long time ago, but we have looked at these reports in detail before, So I'm not going to open the FastQC reports. I'm just going to run MultiQC, and we'll have a quick look at the MultiQC report. So I'm just going to, while this is just finishing, I'm just going to find the appropriate place in Finder. Okay. So FastQC is very nearly done. And after FastQC is complete, there we go. 1 second, bear with me. Just need to find the right, there we go. Okay, so let's bring that back up. There we go, so FastQC is finished. So now we're going to run MultiQC. And MultiQC takes an argument that is an input directory, and it looks in that directory for any relevant report so that it can assimilate into its own MultiQC report. Okay, so we're going to run MultiQC with one argument, which is the current working directory. MultiQC will then look in that directory for any appropriate files that it can assimilate into its MultiQC report. So it should find 2 reports, and it has indeed found 2 FastQC reports. So those are these HTML files that FastQC produced here. There's one there, there's another one there. So those are the 2 reports that MultiQC then assimilated into its own report file, that is multiqc.html, that file there. So we're going to open that file in our browser. And because I share my virtual machine files with my home machine, I can navigate and find that file on my home machine. So here's my share directory on my MacBook. We're in week 10 here, and so in week 10 we have the MultiQC report file, multiqc report.html, which I'm going to open in my browser, and then I will pull that up for you. There we go. So here is the MultiQC report, and we can see that we've got, as I said before, about 1.4 million sequences in each of our in each of these files, in each of these 2 files. We can add in a column here to tell us what the average sequence length is in each of those, so just over 200 bases. Here we've got the counts, so this is the number of reads in each, and it's highlighted in this bar plot the number of duplicate reads in each of those sequences, in each of those files, so around about 75% unique, 25% duplicate, which is fine. Okay, our forward reads, so our first in pair reads, the _1 file has quite good quality across the board. The _2, so the second in pair reads, the quality is still pretty good in the main. It's consistently lower than in the first in pair reads, and it does drop down into this kind of orange zone towards the end of the reads. This is very typical for MiSeq sequencing. You quite often see with this the MiSeq chemistry, especially with these very long reads. So these are over 200 cycles on the sequencer for each of these reads, that the second in pair you will quite often find is slightly lower quality than the first in pair. I don't personally think there's anything particularly problematic about the quality here. So even at base 250, the average is, the median is still kind of 200, still sort of about 24. So although it's lower quality, it's not the total end of the world. So you can see the average quality per read is down in the. We've got a little bit of bias in the beginning of the reads. That's probably due to, if this is genome sequencing, which I think the SRA entry, which I've got open here, suggests that it's whole genome MiSeq sequencing. It's using this Nextera XT library prep. So I would suggest that the way that the genome has been fragmented is enzymatically by tagmentation, and so therefore the bias that we're seeing here is due to that bias in that enzymatic process, giving you slightly biased base compositions at the beginning and the end of the fragments. GC content, we've got this little uptick down here, which is slightly unusual, but otherwise you're seeing a reasonable distribution of GC content around about this center point of about 53% GC. What's the GC content of E. coli? Should be in this entry actually. 50.8, so possibly slightly higher than we would expect, but not beyond the realms of possibility. Okay, so that's that. And obviously that peak is slightly off to the left, but 51% is pretty close to the center of that peak. Okay, per base N, yeah, not massively worrying, nothing to be too concerned about there. Some N's early on in the reads, but nothing too dramatic. Sequence length distribution, most of our reads are 250 bases long, or 251 bases, and that distribution is very heavily right skewed, which is what we would expect. Duplication levels relatively low. For genome sequencing experiment, this is pretty good. And so overall, no adapters, so that's good. So overall, this is a pretty decent quality sequencing experiment, so that's quite nice to see. Although I've picked this this experiment very much at random, what we're seeing in the results from FastQC here is that this is okay. There's nothing dramatically bad about this sequencing. Okay, so that was our first process, was to have a look at that, have a look at the QC, because it's always important to do that. I wouldn't necessarily want to proceed at this point if that QC was terrible, but the QC is fine, so we're going to carry on and we're going to produce our alignments. So the first thing that we need to do, if you remember, I said this was a two-step process. The first step of which was to use this Burrows-Wheeler transform to index our genome to produce what is termed by the Bowtie authors as an FM index of our genome. And to do that, we need to index our genomic DNA FastA file that we downloaded from the NCBI, this file here. I just want to be absolutely sure that this is going to work first time. This is, at the moment, this is compressed using gzip compression. I'm going to uncompress it just to make sure that Bowtie2 can read it. So I'm going to use the Linux program gunzip or gunzip to undo the gzip compression on our genome file. So that just takes one argument, which is the file which you want to uncompress. So we're going to uncompress our genome using gunzip, and you will see that that has changed our FNA file, so sorry, fna.gzip file to an FNA file. So FNA meaning this is a FastA file for a nucleotide sequence. That's where the N comes from in there. So that's our reference genome, and we're going to index that using the binary which I mentioned before, Bowtie2 build. Okay, so Bowtie2 build is the binary for making our genome index. And as with a lot of bioinformatics software, it has a help file. We can have a look at that help file. I'm just going to pipe it into less so we can look at it a page at a time. So this is the help file for Bowtie2 build. At the top we have the usage. This is the synopsis. So Bowtie2 build. We've got some options, which obviously are optional. So we can, those options are described in the remainder of the help file down here. But we also have two arguments that we have to give it. The first one is the reference in. That is the reference file. That's our FastA. That's our FastA sequence file. So that's a comma separated list of files with reference sequences. So that's our reference sequence. And then the second is what's called the BT2 index base. And this says write BT2 data to files with this directory or base name. So this is essentially a prefix that Bowtie2 will use to write all of its output files to. So I'll show you how that works in a minute. And then there are all of these available options for Bowtie2 build, and they're described here. And for most cases, you don't necessarily need any of these options, not immediately anyway. So I'm not proposing that we're not going to add any of these options to this particular command. We're just going to provide these two required arguments. That's our reference genome file and a prefix which Bowtie2 is going to use to write our genome index to. Okay, so here we go then, Bowtie2 build. So it's Bowtie2, then hyphen, and then build, all one word, no spaces. That's the executable that we're going to run. Then we're going to point it to our reference genome FastA file. So that is the file that we downloaded and just uncompressed. So Bowtie2 build gcf underscore blah 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 .fna. And then the second argument was a prefix for Bowtie to write the resulting index files into, and I'm going to call our index here, I'm going to call it E. coli underscore K12. Okay, so that is my prefix for writing out my index. So I'm going to run this. While I run it, I'm going to answer Bernard's question about trimming. So off it goes. It shouldn't take very long actually, but it's going to splurge a load of stuff to the screen. But while that's running, so can you rescue an experiment by quality trimming if the experiment was not perfectly performed? And my plan to do this while Bowtie2 build was running is scuppered by the fact that it's finished in 2 seconds, but never mind. So the answer to that is kind of yes and no. If there's something went wrong with the sequencing such that the quality drops off really, really badly. So if we have a look at our MultiQC report again, where we see quality drop off, especially in the second impair reads here. If this really dropped off really dramatically down into this red zone, and maybe even much earlier in the read, where you have this something as the sequencing hasn't gone exactly as you really planned it to, and there's been a problem, you can use tools to trim reads based on the quality of the calls. So what the quality means here in a sequencing experiment is it's related to the probability of the base caller having made an error. So it's the Q value, which is the quality score, is the minus log 10 probability of the base caller being wrong. So the lower that number gets, the more likely it is that the base called at that position is not actually the base that was present in that sequencing fragment. And that's obviously going to be a source for error in any downstream processing that you do. If you've got the wrong base call, then alignment is going to be more difficult, and a lot of downstream processes which rely on that accurate alignment are also going to be more difficult. So can you rescue that experiment by performing quality trimming? Well, you can certainly help it out. You can get rid of those poor quality base calls, those base calls that are more likely to be wrong, and therefore that will help your mapping. Your mapping is more likely to be accurate, and the bases which map are more likely to be correct. But that can cause other problems. So it can, obviously you plan your sequencing coverage based on the number of reads that you're going to generate and the length of those reads. And if you're suddenly truncating the length of those reads dramatically by quality trimming, then you may not have the coverage in your sequencing experiment that you expected to have. And that can cause other problems. So you can't 100% rescue an experiment. And quality trimming isn't always the right thing to do, but it can really help take data that you may not have been able to analyze at all and render it sort of analyzable by quality trimming. So it can absolutely help you to analyze an experiment that otherwise may have been kind of refractory to analysis. Okay, so that explanation obviously took a lot longer than Bowtie2 build took to run. But let's have a look at what Bowtie2 build did. So it ran very quickly. It produced an awful lot of output, which is really quite incomprehensible, this kind of log output that is produced. But more importantly for our purposes here, it's produced a few files. It's actually produced 6 files here on our hard drive. So we've got E. coli they all begin with E. coli underscore K12, which was that prefix that I supplied to my Bowtie2 build argument. So Bowtie2 build, the second argument was this prefix, E. coli underscore K12, and all of my 6 files begin with that E. coli underscore K12. And we've got 6 files, so .1, .bt2, .2, .bt2, .3.bt2 and .4.bt2, and then rev1 and rev2. So this is our index. This is our Burrows-Wheeler transformed representation of our index, which is stored in several files because there's various aspects of that index that we want to store, and so they're being stored in these different places. So that is our index. It's also stored as binary, so it's not plain text. We can't read that index, but that is our genome index, and that's the thing to which we are going to align our reads. Okay, and so the alignment then is done using a second binary. So when we installed Bowtie2, we actually installed several programs, one of which was Bowtie2 build. The second of which that we're going to use is the aligner itself, and that is Bowtie2. So Bowtie2 is an extremely widely used aligner for short read sequencing data, and it has a help file, so we can find out the help by running Bowtie2 --help. And again, I'm going to pipe it into a pager because Bowtie2 is a very configurable piece of software. It has lots and lots of options, and those options are explained in the help file in a relatively large amount of detail. So the help file is quite long, so you end up with lots and lots of pages of information. So here is the Bowtie2 help file, which again at the top you get the usage, and this tells you, if you know how to read it, this tells you how to use Bowtie2, the things that are required and the things that are optional. So what's required is an index, so that's usually specified with this -x and then the name of your index, and the name of your index here is the name that you gave it when you built it. So that's the prefix that we used when we created our index. So that's the name of our index. Okay, so that's the first thing that we need. The second thing that you need is something to align. Input is what you're going to align to, so that's your genome index. The second piece of input is what you're going to align to that index, and that's our reads. And there are four different ways, of providing the reads. So you can provide mate pairs, so -1 and -2, to give you a paired end sequencing experiment. So this is what we have. So this is what our input is going to look like. -1 and then our first in pair reads, -2 and then our second in pair reads. Or if you've got a single end experiment, then you can provide it as -u for unpaired. Or, and you've got this pipe here is saying or. So paired end reads, or unpaired reads, or paired end reads in interleaved format. So that's where you've got paired end reads, but they're present in the one file. So you've got your first in pair read is the equivalent second in pair read is the second read, and then your second pair comes as the next set of 2 reads, and then so on. So that's interleaved reads. Or you can provide it an already aligned file. So BAM allows you to provide unaligned BAM as your input, and we'll talk about BAM and we'll talk about alignment format and about BAM and SAM in the next session on Friday. So that's your input, one of those 4 things, and we're going to be providing pairs, so we're going to be providing -1 and -2. And then finally you've got optional output. So -s lets you write the output into what's called a SAM file. So that's a sequence alignment map file, so SAM for short. So SAM is a standard alignment format, which, as I said, we will talk about in more detail in the next session, in number 45. By default, Bowtie2 will produce its output to standard output, so it will print its output to the screen. And that can be quite useful, and lots of well-written bioinformatics software will produce its output by default to standard out. There's also lots of software that doesn't do that, especially software that produces multiple files. Like Bowtie2 Build, for instance, had to produce multiple files. So it couldn't produce that output to standard output because you can't separate the output streams then. But where you're producing one artifact of output, so in this case we're producing an alignment, that alignment is produced by default to standard output because that allows us to redirect that output stream into other tools should we want to. And we can look at examples of doing that later on. But today we're going to produce the output into a SAM file. So we're going to define this -s argument at the end. So these are all of our required. So these things here are required. That's the inputs. And Bowtie2 won't run without the appropriate inputs. But we also require some sort of output. And that will either be standard out, or in this case, we're going to define the output using this -s argument. But we also have options. So we can also define a set of options for Bowtie, which will change the behavior in the same way as options for the other command line tools that we've talked about recently, or talked about on this course, change the behavior of the command line tools. We can do the same thing with Bowtie2. We can provide these options to change the behavior of Bowtie2. And there are lots and lots of options for Bowtie2. So we can scroll down this help file and there are loads and loads of options here. And what I'm going to do in the next, so today I'm going to produce an alignment. I'm going to do that using the default mode of operation. So I'm not going to provide any options to Bowtie2 right now. But in the next session on Friday, if you're watching live, I am going to have a little bit of a look at some of the parameter space of Bowtie2. So we're going to do a little bit of exploration of what some of these options do. So that's the plan for Friday, is to look in a little bit more detail at how we can change the behavior of Bowtie2 to perform particular tasks, what difference that makes to the output, and we're also going to look at that output in a bit more detail as well. So we're going to look at SAM files and at BAM files and see what they look like. Okay, but right now, what we're going to do is we're going to produce an alignment. So to produce an alignment, we run Bowtie2 with our three, it's actually four because ped and data, but our three blocks of the three arguments that we need to provide. So two of those are input. So the first one is our reference genome, which we provide with the -x option. So our reference genome is what we created with Bowtie2 Build, and we gave it a name, and the name we gave it was E. coli _K12. That was the name that we gave our Bowtie2 index when we built it using Bowtie2 Build on this command that you can see a couple of lines above. So this tells Bowtie2 where to find these 6 files that make up the index. Okay, so that's our index, and Bowtie2 knows to add the .1, .bt2, .2, .bt2, etc. to actually find all of the separate files for that index. So we just give it the name. So then the second input, that's the genome that we're aligning to, and then we want the thing that we're going to align to that genome, and that's our paired end reads. So these are defined with the -1 and -2 options. So -1 is our first in pair reads. So that's SRR 1186676 _1.fastq.gz. Okay, that's our first in pair reads. And then because this is a paired end experiment, we also need the second in pair reads, and that is SRR11866736 underscore 2.fastq.gz. That's our second in pair read. Okay, so that's our inputs. So our reference genome is the one input, and our reads that we want to align to that reference genome, that's our second input. And in this case, because it's paired end reads, it's actually our second and third input. And then the final thing that we need to define for Bowtie2 to work as we expect is the output. And we want to write the output to a SAM file. So we're going to call our SAM file after the name of the experiment. So SRR11866736 dot SAM. Okay, so that is our output file, which is going to be a SAM file. We'll look at the SAM file in more detail tomorrow. Okay, I gun-zipped the reference genome, but I did not gun-zip the reads because I know that Bowtie2 can understand the gzip compression, so I didn't need to gun-zip the reads, so they are still gzipped. Okay, so that's our Bowtie2 command, and this will take those 1.4 million pairs of reads, and it will align them to our E. coli reference genome, one pair at a time. And that sounds like it will take ages, but actually I think what you'll see is that even on a single core, which is this will run on one thread on a virtual machine, this will be relatively quick. This shouldn't take a huge amount of time. So I'm going to run this. It's going to take over my terminal for a few minutes, but it shouldn't take a huge amount of time. And once it's aligned those 1.4 million pairs of reads, we'll get a little report about how well the alignment has performed. So we'll get a little bit of output, which is actually standard error, not standard output, at the end of this process. So to standard error, we'll get a little alignment report printed once the alignment has actually run. So while that's running, I'm just going to quickly show you a tool called Top. Top allows you to monitor processes on a Linux machine. So we can see our Bowtie alignment process running here at the top of our output on top. So this is updating every couple of seconds to show me what's happening. So this is my Bowtie2 alignment process running here. You can see it's using about 85% of a CPU, more or less, changing over time. And it's been running for at the moment about 54 seconds. So this is updating every couple of seconds and showing me that that process is running, it's consuming CPU, and there's a whole load of summary information on here as well about what my system is up to at the moment. So how much CPU it's consuming, the percentage of the total CPU that's being consumed, the amount of the memory that's available that's being consumed by my machine, and various other bits and pieces of information on here. So Top can be quite useful for monitoring your system if you think that anything is working particularly hard, or that a process has been running for a long time and you're wondering if it's actually doing any work. Top can be quite useful for looking at what your system is up to at the moment. So what is consuming your CPU and what's consuming your memory. So you can see that my Bowtie process is at the moment consuming 90% of my CPU and less than 1% of my memory. So it's really, Bowtie2 is really quite memory efficient. It's one of the reasons why I'm using it as the aligner today is that it really doesn't consume a huge amount of memory. This is taking slightly longer than I expected it to. Maybe 1.4 million reads on a VM is slightly more of a challenge than I thought it was. But it's whirring away, and it's going to produce an alignment, and it's not going to take like an unreasonable amount of need to stop this now and show you the output, show you the little alignment report tomorrow. I'm going to keep mugging to the camera for you until this is finished. So lucky you get to hear my voice rambling on for another few minutes while it's running. So we're still going, 2.5 minutes in now, this is producing an alignment. Actually, we can maybe have a look while we're going at the alignment as well. So that's another useful tip here. Function of the tail command is as if a file is being produced, actively being produced, you can use tail with the -F option, capital F, to look at the end of the file in kind of live as it's being produced. So we're producing a file which is srr blah 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 .sam. If we use tail -F while that file is being produced, we can look at the end of that file dynamically. And actually this is the point where I am going to demur and I'm going to decide to call this a day, because I can see from tail here that I know we've got 1.4 million pairs of reads, and at the moment Bowtie2 has managed to align about 240,000 of them. So actually we're only about a fifth of the way through the file. So maybe I am actually going to call this a day. Dan's asked me to talk a little bit about threads. So threads is a way, so you may have heard of parallel computing or multi-threaded computation. You can run Bowtie2, and Bowtie2 is one example of a tool that you can do this on multiple CPU threads if you have them available. So if you have a machine with a dual core or a quad core processor, you can run your alignment on many threads at once, many threads of computation at once. You will achieve some benefit, some speed benefit from running your software multi-threaded. So Bowtie2 is actually an example of a tool that parallelizes really, really well. So there's an option for Bowtie2 which is -P or --threads, which allows you to run it on multiple compute cores if you have them. My virtual machine, I think, only has one CPU available to it, so I couldn't run this on multiple cores on my VM even if I wanted to. But I have systems, so my unit's workhorse server, which we use for a lot of our work, has 80 threads, has 80 CPU threads available. So I could run Bowtie on 40 threads on that machine, and it wouldn't quite go 40 times faster, but it would go quite a lot faster than just running it on a single thread. So it allows you, if you think about what Bowtie2 is doing here, it's taking a pair of reads and it's looking for an alignment of that pair of reads to the reference genome. And that alignment problem for that one pair of reads is independent to the alignment problem of all of the other pairs of reads. So you can run that alignment process for multiple reads simultaneously because they're all independent of one another. And that's what the parallelization does for Bowtie. It takes those 1.4 million separate alignment problems and it splits them up among the available threads. So you can run 40 of, if you've got 40 threads, you can run 40 of those alignment problems simultaneously. And in theory, you can solve the whole problem, the 1.4 million alignments, 40 times faster by doing that. It doesn't actually work out as being 40 times faster because of various little bits of overheads and things. But that's the idea behind running multiple threads for a program like Bowtie2, is you can take advantage of having multiple CPU cores available on your machine to parallelize a particular job. Okay, because this is clearly going to take another 5 or 10 minutes, I am at this point going to sign off. So I will leave this running and I will leave it at the output, at the standard error output of Bowtie2 once it's finished for session number 45 for the next time we meet again. So so with that, I will sign off and I will call it a day. Thanks very much for tuning in. Keep safe and stay well, and I will see you all next time for number 45, where we will look at the output of Bowtie2 and we'll look a little bit about the realization of Bowtie2. Okay, so thanks everybody and see you next time. Bye.